we're going to look at the Saper paper in this video. Remember that the Saper paper is itself a discussion of another paper that was published in the same journal, the same issue of the same journal. So the Saper paper makes the statement, they produced transgenic mice in which expression of the UCP gene, so that's the uncoupler gene we talked about in class, let me write that down, this is the uncoupler gene, is placed under control of a promoter for hypocretins, otherwise known as orexins. So what is a promoter? The promoter is a regulatory region. So promoter is another way of saying regulatory region. Okay, so orexins are peptides that are produced only by cells in the lateral hypothalamus. By placing UCP expression under the control of this regulatory region, the investigators effectively placed a small heater in the hypothalamus. So, the regulatory region is the part of a gene that determines when and where gene expression occurs. And the coding region is the part of a gene that determines what protein is expressed. Okay, so the uncoupler protein, so uncoupler is in red there, is being created wherever the right conditions are for this regulatory region to be activated. And in order to do that, there are proteins in only some cells. Those proteins are called transcription factors. Only some cells will have the transcription factors that will activate this regulatory region. And those are only going to be found in parts of the hypothalamus for this particular regulatory region. So we get this protein being made because of its regulatory region attachment in the hypothalamus. And so we know that an uncoupler protein is indeed a molecular heater, so far so good, nothing controversial. A little later in the paper it says that the result is that the transgenic animal expressing the uncoupler gene had a continuous reduction in body temperature, about 0.3 to 0.5 degrees centigrade. Now let's look at the data from the Conti paper itself to see how well that statement is supported. So males that are wild type are in black. Males that are engineered, this is the engineered version. So they've got the uncoupler protein in them. Notice that the engineered version is in red then. And sure enough, you see some periods of time when it's the red data seems to indicate a reduced temperature when compared to the wild type data. Let's look at our axes, time of day, and let's look at their y-axis there, the temperature. Okay, so I've made a line across this graph to show where 37 degrees is. So that's the temperature around which the body temperature of this animal seems to vary. What are these stars here? These stars are the only points that the data that they gathered, when put through a statistical analysis, is significant. That means that so far as you can tell from this data set, there's only a few times of day in which the temperature of the engineered animal was actually different than the temperature of wild-type animals. That line under that star means that that whole region of time has significant differences between the wild-type and the engineered mice. But okay, so what the paper says is a little inaccurate. What they really should have said is that there are In the light, that is not a relationship that's true at all. In the light, there's no difference whatsoever between the wild type and the engineered animals. What's different about these animals between light and day? So I told you, orexin is a gene that is expressed when the animal is awake. And in fact, orexin is a gene that if you're missing it, or if you have a copy of it that's not working in some way, you might become narcoleptic, which means you fall asleep at very strange times. Okay, so it's expressed, this gene, only in the hypothalamus, and it's expressed only when you're awake. When do you think mice are awake? Must be when there's a difference between the wild type and the engineered creature, huh? Oh yeah, mice, therefore, can be seen to be nocturnal. Okay, what else do we learn from this? Well, that's about it. Okay, now let's go to another statement in the paper and another set of data that's being graphically represented. So, says, in the experiments, the hypothalamic transgenic mice showed no change in food intake or physical activity, but their body weight did increase. All right, so let's take a look at the data. Yeah, there doesn't seem to be a little variation, but no significant differences, maybe with this one little weird exception here, which they choose to ignore, and I think that's good judgment. No difference in locomotor counts. What's that? So this is really an measurement of the activity of the animal. And then daily food intake. Well, are red and black lines different? Remember, the black lines, the black bars in this case, are the wild type, and then this is the engineered again. So wild type and engineered. No significant difference. No significant difference. No significant difference. So what they're telling us is that the daily food intake is the same between the wild type and the engineered mice, and it looks to be true. Then they say, but the body weight, here's the body weight, y-axis here, age is presented as the body weight is presented as a function of age, and at least among the males, it's a little more subtle among the females there. It's the females. But within the males, there is a difference, and they claim in the body of the paper that this is a significant difference. You can see that it's standard error bars, whether they're standard deviation or standard error, do not overlap, but other types of analysis were carried out, and this is apparently a significant difference in body temperature, way out when these animals are quite old. And you can see that throughout a lot of their lifespan, it does seem to be a pretty good trend that the male animals are considerably bigger when they're engineered than the wild type. Okay, and then they say, presumably due to, and you can ignore the presumably due to part, because what these authors presumed was that the basal metabolic rate of these animals was different. Now, they themselves went on to carry out experiments to demonstrate that that wasn't true. So the engineered mice have the same basal metabolic rate as do wild-type mice. So your question, I'm not going to answer this question. The question I'm going to ask in class is, why are they gaining weight?